So in my lab, we have been interested for many years in the mechanisms of pain and temperature sensing, and there are many of them, right? So pain can be triggered by strong mechanical stimulation, by extreme temperatures, such as noxious heat or noxious cold, and even by irritant chemicals, such as capsaicin, which is the active ingredient in chili peppers. Paradoxically, pain can also be triggered by light. That's not something that happens every day, but there are some genetic disorders, fortunately very rare, in which patients suffer from exposure to light. And we have been studying those mechanisms for some years. And finally, in more recent times, we also developed an interest in itch mechanism, particularly serotonergic itch, with focus on that as well. But that's just the overview, because the topic today is about statins. So if you have high cholesterol level, of course you're in trouble, because you get atherosclerotic disease, and the WHO tells us that this is associated with high mortality rates. But fortunately, we have statins. So statins are something like magic bullets. They're really effective in lowering cholesterol. I've shown here some of the most widely prescribed statins, like simvastatin, atorvastatin, and rosuvastatin. It is estimated that perhaps 250 million people in the world are prescribed daily statins. How do they work? So statins block the first step in this pathway, in this cholesterol biosynthetic pathway. But as you can see, the pathway is really complex. It's not just about cholesterol. So we should not be surprised that statins also have what we call pleiotropic effects. That means biological effects which are independent from their cholesterol lowering activity. Right. So the starting point of this study was that we were puzzled by what we found in the literature, because on the one hand, if you look at the clinical studies that were done on patients, it has been published that even though very rarely, statins can have adverse effects. So they are associated with increased probability of myalgia, muscle pain, and even painful peripheral neuropathy. While on the other hand, when you look in the literature and you read about animal models of chronic pain, here mostly beneficial effects of statins are reported, including analgesic and anti-inflammatory action. So how to reconcile these two sides of the story? We decided to look at that. So this is, in principle, a very simplified cartoon of a peripheral nerve ending, nociceptive nerve ending, and as you can see, we have two types of channels mainly. So on the one hand, on the left, we have what is called transducer channels, which are activated by specific stimuli, and here on the right, we have amplifying channels. These are voltage-gated sodium channels, which are activated by changes in membrane potential and trigger the action potential. So what happens here is that transducer channels, and some of these channels I'll show you are members of the Transient Receptor Potential family. So some of these channels are activated by extreme heat, by noxious heat, by cold, by chemicals that shown, and by mechanical stimuli. They open, they allow cations to enter, mostly sodium, but also some calcium, and this triggers what is called generator potential. That's a membrane depolarization, which, if it's large enough, it crosses the threshold, and it activates voltage-gated sodium channels, which are now involved in triggering the action potentials, and these are the signals that are propagated all along the peripheral axis of these neurons to the spinal cord, to the CNS, central nervous system. And that produces pain. So in order to have pain perception, you need a conserved activity of both types of channels. Right, so the main features, the main characters in our story will be TRPV1, which is a receptor activated by capsaicin and by noxious heat, TRPM8, which is the cold and menthol receptor, and TRPA1, which is activated by many types of irritant compounds, including allicin, which is the active compound in onions and garlics, cinnamon oil, mustard oil, and also by it's also mechanogated, so it contributes to mechanically induced pain. More recently, we also started to become interested in the sodium channels themselves. In those receptors, we have a combination of NAD1.7, which is a TTX sensitive variant, and NAD1.8, which is a TTX resistant variant. How do we investigate these channels? We can do two things. On the one hand, we can culture DRV neurons from rats and mice, mostly mice recently. We keep them in primary culture. On the other hand, we also have the heterologous expression system, where we can express all these channels that I mentioned, including many mutants. And then we use functional techniques such as calcium microfluorimetry. We look at calcium transients in the cells. and also patch clamp. So we do all varieties of patch clamp, the voltage clamp, the current clamp, all this depends. So what we see, this is the data. We notice that simvastatin activates both TRPV1 and TRPA1. I'm talking about human channels reconstituted in HEC cells. As you can see, two applications of simvastatin activate produce calcium transients reproducibly. The same here for TRPA1 expressing cells. And these responses disappear when we remove and take away external calcium, showing that we're dealing with calcium entry and not with calcium release from stores. And finally, we also saw that these responses are completely abolished in the presence of selective antagonists for either TRPV1, this AMG compound, or TRPA1, which is A96. That was not very surprising because I mentioned almost everything that you throw at TRPA1, for example, will activate. We used to call it, when I was working in airline in Germany, we used to call it the sigma voltage receptor. So it's basically very, very promiscuous. But we were more surprised in our finding that rosuvastatin acts as a selective agonist of TRPM8, the cold and menthol. Similarly, as I've shown, two applications of rosuvastatin activate calcium transients in cells expressing TRPM8. We validate the expression by the response to the selective agonist published, well-known agonist WS12. And again, as in the case of TRPV1 and TRPA1, the response is abolished in calcium-free conditions, and it is blocked entirely by the selective TRPM8 antagonist AMTB. These effects are dependent on the concentration of statins. This is the case of TRPV1, the case of TRPV1 for simvastatin, similar EC50s around 20 micromolar. In the case of TRPM8, we used two methods just to make sure because it was such a surprising discovery. So we used both patch clamp on the upper trace here and calcium imaging, and they both yielded reasonably similar values for EC50, a bit higher than the case of simvastatin, so close to 100 micromolar. We also wanted to investigate the molecular determinants. So where exactly and how exactly these molecules activate the channels. So first, in the case of TRPV1, we expressed a mutant. This mutant lacks three cysteines, two located in the N terminus, one located in the C terminus. And this mutant is insensitive to reactive oxidant species. TRPV1 is known to be potentiated by oxidative stress. And in our hands, this mutant indeed showed a reduced response to simvastatin. More intriguingly, we expressed the capsaicin sensitive TRPV1 mutant. The binding spot is around here. And what we saw is that whereas the mutant could not be activated by capsaicin, it could still be activated by low pH, because this channel is multimodal, responds also to acidity, but it shows no response to simvastatin. So again, the action of simvastatin seems to require an intact capsaicin binding site. So we're still not clear what this tells us, whether this is a combination of factors or what is more important. In the case of TRPA1, similar to what we've seen before, we expressed another mutant which lacks three cysteines actually, three cysteines are substituted, of course, with serines in the N terminus, and again, this mutant is insensitive to reactive oxidant species and also to electrophilic agonist of the channel. And again, this mutant seems to be defective in its response to simvastatin. It's statistically significant. Finally, in the case of TRPM8, we have a wild type which responds nicely to rosuvastatin and to menthol, and also to a synthetic agonist to isolin. The menthol insensitive mutant could respond perfectly to rosuvastatin. I'm not showing that here. But we have another mutant which is insensitive to isolin. It requires this aspartate here in position 802. And this mutant presented a reduced sensitivity to rosuvastatin. This location is here in the third transmembrane domain. So we're pretty sure that we are close to identifying the molecular determinants. And now, of course, the thing is to go to the native system, to go to the real thing. And the real thing, of course, would be nerve endings. Ideally, one should study these channels and these receptors where they belong, the nerve endings themselves. But this is the problem because these nerve endings are very fragile, very tiny, they're very difficult to extract and isolate. So we have a solution around that. The solution is to express the soma of the DRG neuron as a model of its own terminus. So it goes like this. Normally, the cell body physiological condition will express also proteins like transducer channel, sodium channel, our receptors, and they each are sorted and trafficked where they belong, where they do their work. However, in culture, when you cut the axons of the neurons at both central and peripheral axons, you end up with a soma, and the soma still makes its channel, but now they have nowhere to go, so they start being expressed in the membrane of the soma. And there are advantages because in the soma you can do a lot of things. You can do returns, the nerve ending, you can do patch mapping, you can do calcium imaging, you can do a lot of things. Of course, there are caveats because it's not the real thing, it's not the terminus, it's a model. Many changes can occur in culture, depending on the growth factors that you have. So we consider this to be sort of a hypothesis generating system, a good model for the terminus. So this is how they look. These are neurons in phase contrast. They're 2 or 3 days old, so they start growing processes in culture. So the question is, what happens when you apply simvastatin to such a culture? Well, you'll see here, have a look at the left hand. This is the same optical field in fluorescence, in epifluorescence. And while simvastatin is being applied, you see one of the neurons responds, while the other does not. And this can be quantified. So we see responses to simvastatin. We were very pleased. The effect can be reconstituted in the culture ERG neurons. And we can also do pharmacology. So these are examples of neurons activated by simvastatin applied twice, and then followed by a selective agonist of TRPA1 and another one, TRPV1. What we see is that all neurons that are sensitive to simvastatin are also activated by either, or they also express either TRPA1 or TRPV1 or both, which correlates well with the pharmacology that we've seen in PET cells. And again, these responses, these calcium transients in neurons can be blocked to some extent by the selective TRPV1 antagonist, by the selective TRPA1 antagonist, and even more potently by both. They have a cocktail containing both antagonists, and the response is completely gone. And similar things happen to rosuvastatin. It activates a subpopulation of neurons, and most of them will also respond to the selective TRPA8 agonist, WS12. And again, when we apply the antagonist, AMTB, it almost completely and irreversibly, as it seems, blocks the response to rosuvastatin. So we are pretty confident that the DRG neuron sensitivity in culture to simvastatin is entirely accounted for by the expression of TRPA1 and TRPV1, whereas the responses to rosuvastatin are accounted for by TRPM8 expression. This is recent, the stuff that I've shown you was published recently. This is unpublished data. So as I mentioned, we also started to work on sodium channels. So here we have 3 different sodium channels. We have the TDX resistant NF1.8, which is expressed in those receptors. We have TDX sensitive sodium channels natively expressed in this cell line, and also expressed the cardiac sodium channel. And all of them, interestingly, could be inhibited this time, right? So not as in with TRIP channels where we have an agonistic effect. This time we have an inhibition, concentration-dependent inhibition with an IC50 of around 5 micromolar. In all cases, this shows what is called a use-dependent block. So the more you activate the high frequency in the channel, the more potent the inhibition becomes. This is also has been shown with local anesthetics, so it's quite similar. So yeah, on the one hand, to summarize this part of the block, what we see is that some statins are pro-analgesic, so they activate TRPA1 and TRPV1, which are known to trigger pain. They are involved in pain signaling. But on the other hand, rosuvastatin activates TRPM8, and activation of TRPM8 leads to analgesia. That's interesting. And on the other hand, simvastatin also blocks, to some extent, sodium channels. So these effects will counter pain, they will be analgesic. Right, so have we solved the puzzle that we started with? No, not really, but I think we're making some steps. So the activation of the transducer channels of TRPA1 and TRPV1 could be involved in myalgia, possibly neuropathic pain, whereas the other effects may, in other conditions, may lead to a decreased pain perception. And now to the second part, because I advertised at the beginning that I'm talking about pharmacology, but also modulation. Let's talk a bit about modulation. So this has to do with inflammation. So inflammation, what we have, the tissue division, we have recruitment of immune cells, which start releasing also the pro-inflammatory factors like NGF, mast cells granulate, releasing histamine and proteases. Platelets release serotonin and other factors. Of course, bradykinin is also produced. And finally, the tissue itself. The damaged cells start producing a sort of phospholipid mediators, including prostaglandin and prostacyclin. So to focus on that, the prostacyclin is a pro-analgesic or pro-inflammatory mediator. What we also know is that the nerves themselves, so nerve endings themselves, they contribute to the inflammatory process by releasing neuropeptides like substance P, producing plasma extravasation, edema, and CGRP, which produces vasodilation. So this constitutes what called neurogenic inflammation. So it's a complex story, but let's focus a bit on prostaglandin and closely prostacyclin. Sorry. What we wanted to look at was the interaction between prostacyclin, which is a pro-analgesic pro-inflammatory mediator, and TRPM8, our cold and menthol receptor, which is known to be anti-inflammatory. Do they interact functionally in any way? So first we expressed the IP receptor for prostacyclin and TRPM8 in HEX cells, and we applied cold ramps. So these are CR cold ramps. So this is a controlled experiment in which we applied cold ramps twice. Temperature goes from about 32 to about 22 degrees in less than 60 seconds. Quite reproducible. And then when we applied in between the two ramps, when we applied chicaprost, that is a selective agonist of the IP receptor, we see a pronounced reduction in the response to the cold ramp. Right, this statistic. So this is an inhibition basically of TRPM8. This is clear. So we first wanted to make sure that this is not a direct effect on the channel. So we used cells expressing just TRPM8, and what we see here, chicaprost does nothing at all. And when you activate the channel with isolin in a concentration-dependent manner, if you add chicaprost to isolin, nothing happens. It's the same thing all over. So the chicaprost has no effect, no direct effect on TRPM8. The effect that's shown before is purely mediated by the activation of the receptor, of its own IP receptor, and it can be reproduced by other synthetic IP receptor agonists like Veraprost and Iloprost. Some of them are used in the clinic. Here we look at the effect of chicaprost at various concentrations on the activation of TRPM8 by isolin, and you see this prominent decrease. This shows that there's a reduced efficiency, not a reduced potency, which is consistent with a non-antagonistic effect, with a non-competitive effect. So it doesn't compete on the same side as isolin. It works differently. What happens? So what's the mechanism? Well, what is known is that the IP receptor couples with the cyclic AMP pathway. This is the canonical part. And indeed, we see that all these agonists, when applied in HEX cells expressing the IP receptor, they show an increase in CRE activity. This is basically important for cyclic AMP concentration. So it induced all these agonists induce an increase in cyclic AMP, which is to be expected. But on the other hand, we also see an increase in calcium. This has been already reported that the IP receptor sometimes can also couple with the PLC pathway, but not always, only rarely. But in this case, it seems to be working now. So first, we thought the culprit of the inhibition of TRPM8 must be the cyclic AMP pathway, but we were surprised to find out that it wasn't the case. So when we applied for scoring, we see no effect on the activation of TRPM8 by isolin. And similarly, so forskolin activates directly at the cyclase, increasing cyclic AMP levels. Nothing happens. We also used choleratoxin. Choleratoxin activates GS, the G protein that activates the cyclase. Again, nothing interesting happens. So it appears that it's not the cyclic AMP pathway. So then we switch gears. We wanted to look at the other one, the PLC pathway. And first, we made sure that indeed it's active, and we have a tool to investigate that. And this YM compound, this is a blocker of GQ proteins. GQ proteins are the ones that activate PLC. And indeed, we see that this compound has no effect on the accumulation of cyclic AMP produced by chicaprost, but it profoundly inhibits the calcium accumulation. So this is a good tool to distinguish between the two pathways. It does nothing to the cyclic AMP pathway, strongly inhibits the PLC pathway. So we went further. So there's a bunch of chemicals which are known to interfere with different steps in the PLC pathway. For example, galline is a G beta gamma inhibitor. U73122 is a PLC inhibitor. PP2 inhibits protein kinases. Nothing worked with one exception. The same YM254 compound was the only one that would restore the sensitivity of TRPM8 to isolin, so it antagonized the effect of chicaprost. And what this does, this is, as I mentioned, it's a direct blocker of GQ. It binds to G alpha Q protein and doesn't allow it to do anything, to trigger the whole pathway further on. We also got our hands on a mutant, a TRPM8 mutant. It's a triple mutant lacking 3 key arginines, which cannot bind G alpha Q. So this mechanism of G alpha Q inhibiting directly TRPM8 has been published before once. And so we got in touch with those people and they sent us a mutant. And indeed, in our case, what we see here, that chicaprost inhibits easily the wild type channel, but it has a much harder time inhibiting the triple mutant. It still does. That's a bit surprising, and still requires 100 times more molecules, but maybe there's something else going on in addition to this direct interaction. Finally, we went back to DRG neurons to make sure that it happens. In DRG neurons, there's not a huge co-expression between TRPM8 and the IP receptor, but there is some. So we were a bit scared that we won't see much, but we were lucky. So this is the challenging DRG neurons with isolin twice, and also with the TRPM8 selective agonist to identify TRPM8 expressing DRG neurons. And as you see, isolin can evoke reproducible responses, no problem here. And then when we apply chicaprost, in many cases, we saw, again, the reduction. So this inhibition, much reduced response to isolin. And this reduction, this inhibition could be reversed, on the one hand, by this compound, which is selective antagonist of the IP receptor, but also by the YM compound that I showed before, so blocker of G alpha. And this is the statistic. Again, this is not so pronounced, but that's only because there's not much of a co-expression between the two receptors. So what we think we see here is that when either prostacyclin or one of the agonists of the IP receptor binds to this G protein coupled receptor, this triggers the cyclic AMP pathway. So alpha S binds to alanine cyclase, producing cyclic AMP, but this is not involved in what we see. On the other hand, we know that the same binding of the agonist activates the PLC pathway, activating G alpha Q that binds to PLC. This triggers, this converts PIP2 into IP3, and IP3 is released to the cytoplasm, activating calcium channels in the ER, releasing calcium. But all these events are, again, not relevant. The only thing that matters, according to our measurements, is this. The direct binding of G alpha Q. It's not a very common mechanism in which a G alpha subunit directly binds to a channel and inhibits this channel. And thereby, this may be the way that prostacyclin produces its pain-promoting actions by removing this brake, by inhibiting TRPM8, which confers protection against pain and inflammation. Right, so these are the people who contribute to the work. We have a fruitful collaboration with my old friend, Professor Andreas Leffler from American University in Hamburg, and the work involving the IP receptor, prostacyclin receptor, TRPM8 was done in a very close collaboration with my friend Sorin Tunau and his group at the Institute of Biochemistry of the Romanian Academy, and I thank you all for your attention.
We have time for questions and comments. Alex, please.
I have okay. Sure. This may be a bit of a question, but I was really wondering how you selected the amino acids substitution sites for the response channel.
No, it's a very legitimate question. So we knew from the literature that some of the sites were involved in different specific functions of the channels, like, for example, redox sensitivity with these channels. In particular, TRPM1 is a very, very sensitive redox sender. So we went for the usual suspects.
Okay.
Just if you can comment a little bit on the possibility how the opening and closing of the calcium channels in the motile, how it's connected with the motility, with the cell movement. Because, okay, I mean, this is the question here, is that because if you start to stimulate some of the channels and how they are, because you get the calcium in or out. So calcium coming from out in would decrease the many kind of adhesion to the matrices or something, but may also stimulate something on the cytoskeleton. Okay? But just coming in, you can also go reverse storage from the blood reticulum. So, but is it anything known about that? Like sort of, or what do you think about it? Is it kind of crazy idea or?
No, for sure. It's not really something that we looked at. These cells are stable. They just adhere to the bottom of the page. They don't move around. These neurons, so they're not motile.
And they affect cells with related transfections and so.
No, they don't move. These are very fast signals. They last a few seconds.
So
It
this
doesn't
is
allow them
okay.
to.
But it's not repetitive, right?
We can trigger responses reproducibly by activating these cells with temperature changes or chemically. And there's also calcium coming in, but neurons they know how to deal with calcium. There are a lot of mechanisms to push the calcium out.
The neurons kind of grow during regeneration, so where they have to.
It surely
Because there
calcium.
are some stories that they would move the cast or in the matrix or on the matrix. That's very important for some growth of the regeneration, and there may be playing the role. But what would be the stimulus to open and close the channel? That's actually really right? And how long it would last, if it has any physiological significance.
For sure, axonal growth is not to be calcium dependent. And interestingly, also trip channels involved in this responses to chemoattractants released by other neurons, by the target neuron. So yeah, it's calcium dependent for sure. But for us, calcium is just a very nice.
I understand.
The signal of neuron activation. It's easy to monitor. Slow your cells, the calcium indicator correctly. Thank you.
Which is the concentration of station?
Well, we can go as low as 100 nanometers, nanomolar, sorry, nanomolar will still activate channel significantly. So it's quite sensitive to capsaicin.
And
And you have to make sure when you work with capsaicin not to touch your eyes.
because it's very hydrophobic. So then you switch from that
Use ethanol.
get on. Is that
And that may explain why we
difference behavior of neurons?
but it's very small volumes. So it dissolves well in ethanol. So ethanol is one part of the 1,000 less.
And the action of capsaicin and simvastatin are synergistic.
No, we didn't look at that, but as I showed, they seem to require the same sites, so they seem to compete with each other. So I'm not sure what we see on the British term, but it's something that we should do.
The effects are not cumulative, or
We don't know.
thank you.